Recording started.
Hi Bio30 students. We had a whiteboard session before the break, and I'm making a second whiteboard session now. And so I would make sure that you've gone through all these whiteboard sessions, these two whiteboard sessions, before our midterm, which is going to be on Wednesday. So again, please practice all the questions on the PDF file that I send. And if you can do all those questions, you should do well on the midterm. So let's jump right into these questions here. And so the first one is talking about a plant breeder trying to breed a true breeding variety of radishes with an oval shape. However, each time he plants with oval radishes were self-fertilized, only half of the offspring were oval. The rest of the plants produced either round or long radishes. So the question says, what would the expected phenotype ratio be if plants with round radishes were crossed with plants with oval radishes? So the first thing I want you guys to notice is the fact that we've got three different types of plants here. We've got oval ones, we've got round ones, and we've got long ones. So this is not your basic sort of dominance recessive sort of pattern where you've either got a tail or no tail sort of thing, or something is short is recessive to dominant. This is a situation where you've got three different possibilities. And so if you've got this sort of situation, that should immediately jump out to your mind that this is not your typical dominance recessive pattern. This is what's called incomplete dominance. And if you can't remember exactly what that's about, please look on page 594. There's an example there about flowers, and it talks about how you can have red flowers crossed with white flowers and get pink flowers. So rather than getting just red or white, you get something that has traits which are in between red and white, and that's called incomplete dominance. So we know that we've got an incomplete dominance pattern going on, and again, what we need to find is the phenotype ratio if round plants are crossed with oval plants. Okay, so again, remember the difference between phenotype and genotype. Genotype's asking you what are the genes actually looking like, whereas phenotype is always what does it actually look like. Are you talking about a round plant, or are you talking about a small one? Are you talking about something that's got curved wings? Okay, those are all examples of genotypes. What are the traits like? Okay, so what we're going to have to do to solve this is to set up a Punnett square that's going to be crossing round by oval plants. Now, to do that, first of all, we need to talk a little bit about what the genes are going to look like when you've got incomplete dominance, because they look different than they do when you're talking about a regular dominance recessive pattern. So let's have a look at that. So on the next screen, I want to show you, first of all, just when the question's talking about each time when you're breeding oval plants by each other, in other words, it says they're self-fertilized, so oval crossed with oval, it says that only half the offspring were oval. Okay, some are round, some are long. Let's see why that is, first of all, by looking at the genes. Okay, so the way it works then is that the oval plants are in between the round and the long, right? If you just imagine round as being like a perfectly round circle, and long is kind of like a very elongated circle, whereas oval's kind of in between the two, right? Okay, so for round, we would call it P with a little hat R. That's the same thing as P, kind of like, if you want to call it an exponent, R, but we just kind of use the hat to represent that, because it's a lot easier doing it that way, especially if you don't have the ability to make those kind of exponents. Okay, so that's what that means. So you can see why the round ones are going to be P with a capital R, and P with a capital R, so it's going to have always two alleles, right? Same thing with long. It's got the L, okay? And because oval's between the two of them, it's going to have PR and PL. So that kind of makes sense, right? If you've got some creature that's got PR and PL, it's going to have traits of both parents. It's going to be in between. Okay, so when we cross the oval, the question says that when they self-fertilize the oval plants, it tells us the results are going to be half oval, and then some are going to be round, some are going to be long. And if you look at the results, when we cross oval times oval, you can see that that's the case, right? Half of them are oval. That's going to be these ones that have both an R and an L, right? This one and this one. And then the other quarter are going to be round and the other quarter are long. Okay, so that's just kind of basically looking at what the question is described, but it's not getting into the actual question, which is asking what happens when you cross round with oval radishes. So let's set up a Punnett square for round crossed with oval and see what happens. Now that we know what the genes look like. Okay, so we're crossing round with oval. So round is 2 R's, oval is 1 R, 1 L. All right, so here's the genes of the parents. We set it up this way for the Punnett square. And so when we do the cross, we get these answers. So you can see the results then when it says, what is the phenotype ratio? Okay, so in other words, what are they going to look like? Well, we can see that these ones are red, or sorry, not red, but round, I should say. So 2 out of the 4 are going to be round, and these 2 are going to be oval, because they've got an R and an L. So we've got 2 round and 2 oval. So you can look at what you think would be the proper ratio for that, 2 round, 2 oval. And what do you think the answer would be? It's obviously got to include both round and oval in it. And what do you think it would be? Okay, so I'll let you guys decide what that would be. And let's move on to the next question then. So the next question says, in guinea pigs, black coat, capital B, is dominant over white B, small b, white coat, small b, sorry, and straight hair, capital S, is dominant over curly hair, small s. Each pair of alleles assort independently. Okay, so it says 2 guinea pigs were crossed. The gametes, in other words, the sex cells produced by the parents, are shown in the Punnett square. So first of all, I want to show you guys something. When you look at this Punnett square here, you're probably looking at it and you're thinking, well, why is it that the one parent has 4 possible alleles where the other parent has only 2 possible alleles? Well, let's figure out why that is. Okay, well, if we look at this parent here, this is the male parent that has these 4 different possibilities. This parent must have had these genes, big B, small b, big S, little s. The reason is, if you look at the results, you can see that some of the genes are big B and some are small b. So the parent must have had both a big B and a little b gene. In the same way, it must have also had big S, little s, because we see big S and little s genes. Okay, now if we look at the mom though, we see here we only have big B and little b, but we have 2 small s's. So the mom must have had big B, little b, and 2 small s's. So because of that, the only possibilities when we look at gametes produced from the mom, we can either get a big B with a little s or a small b with a little s. Those are the only 2 possibilities. So that's why you only see 2 possibilities for the mom. Whereas for the dad, if you're going to grab one of the B's and one of the S's, you can get any one of these 4 possibilities from the dad because there's more things to choose from. Okay, so that's just showing you where the parents' genes came from and why we have a 4x2 Punnett square instead of something like a 2x2 or a 4x4. Okay, now what the question actually says is, what is the phenotypic ratio of the offspring? Okay, so we're crossing the parents and we're finding out what the offspring are going to be. So for instance here we would write big B, and big S, little s. And we could write that down and that would be the first offspring. We do the same for the second and we fill in this whole Punnett square. Once we do that, then we can find our answer. Because our answer is asking us what's the phenotypic ratio of the offspring. So we need to find out how many are black straight, and we would find out how many of the 8 are going to be black straight, and we would write down the number. Say it's 2 or 4 or whatever. Same thing for black curly, and right on down the line. So let's do that. Well, okay, so of course, we'd always want to figure out, what's dominant and what's recessive. So we'd say capital B is black, small b is white. Okay, so we do that first of all for any type of genetics question. And we move on. So here I've actually filled in the table for you. So I've figured out what each one of these individual crosses is. And so to get your answer for your phenotypic ratio, for black straight, we need to figure out how many are black straight. Okay, so we know that black is going to have at least one big B, which is dominant, and it's going to have at least one capital S, which is also dominant for straight hair. So if you look at the ones that have a capital B and a capital S, you have 1, 2, 3, you have 3. Okay? So 3 have black coat and straight hair. So we would write the number 3 in here. Okay? I'm not going to fill all of these in, but I'll show you also how we would get white curly. Okay, white is recessive, so it's going to have 2 little b's. Curly is also recessive, so 2 little s's. So how many situations have 2 little b's, 2 little s's? Just 1. Okay, so we would write the number 1 here. There's only 1 with white coat and curly hair. Okay, so I'll let you guys figure out how many have black curly and how many are white straight, and you can fill these numbers in. And so for your answer, you're going to have a 4-digit number, like 2431, whatever. Okay? So that's how we do that one. Now on to the next one. So the next one says Mendel. Okay, he was a guy who was always working with peas. So he found in peas yellow seeds, which are capital Y, are dominant over green seeds, small y, and round seeds, seed shape, capital R, is dominant over wrinkled seed shape, small r. It says the alleles for these traits are located on independent chromosomes. So it says when this parent and this parent are crossed, what percentage of their offspring are expected to have yellow seed color and wrinkled seed shape? Okay, they're going to have these 2 traits. So it says what the percentage, it asks for what the percentage is, and it says to write this answer as a 2-digit number. So if it was 75%, we would write the number 75. Okay, so we want to know what percent have yellow seed and wrinkled shape. That's what we need to find. So what we need to do is we need to find the probability of having yellow seed color, and we need to find the probability of wrinkled seed shape, and then we need to use the formula for 2 independent events. Now if you think about it this way, finding yellow seed color and finding wrinkled seed shape are 2 independent events, just like if you were to say roll a dice and then flip a coin. Those 2 things are independent. So let's say the chance of rolling a 6 is 1 out of 6. The chance of rolling or flipping a heads is 1/2. So the chance of rolling a 6 and then flipping a heads is 1/6 times 1/2. Because those are 2 independent events, we multiply the 2 probabilities together. So the way that applies to our problem here, the probability of getting round and wrinkled is the probability of round times the probability of wrinkled. Okay, so we always multiply the 2 probabilities together if they're independent events. Okay, so what we're going to do then is separately find out probability of round, or probability of yellow seed, and then probability of wrinkled seed shape. So let's do that. Okay, so first of all, we need to find out what the dominant and recessive genes are. We always want to set that up for any kind of genetics problem. So we do that here. And then we find the probability of yellow seed color. So for this, at this point in the problem, we're going to ignore whether it's round or wrinkled. We forget about those R's altogether and just focus on the Y's. Okay? So even though we figured out that these are the 2 parents, we just focus on the Y part right now. Okay? And so we can see that we've got capital Y, small y, that's going to be this parent here, the vertical, and 2 large Y's, that's going to be this parent. Okay, so that's how we set up our Punnett square. Now we're looking for the probability of yellow seed color, which is the dominant gene, that's the capital Y. If you look at the results we get, you can see we're either getting results that have 2 capital Y's or a capital Y, small y. So in other words, all 4, or 100% of these kids would have yellow seeds. Okay, so the probability of getting yellow seed is 100%. So far we know the probability for the color. Now we need to find the probability for wrinkled seed shape. So again, we know that these are the parents that are crossed, but we just are focusing on the R's. So again, we've got big R, little r. You can see we've got that on the Punnett square, 2 small r's, that's on the Punnett square. And I should have written the results right in the Punnett square, but I wrote them over here. But as you can see, probability of wrinkled seed shape, which was 2 small r's, that was wrinkled seed shape, is 2 out of 4, or 1/2, which is 50%, right? When we're multiplying probabilities, we always put it as a decimal. So that's going to be 0.5. So when we find our final probability then of getting yellow seed shape and wrinkled seed, we multiply the probability of yellow seed shape, which is 100%, and we just call that as a decimal, we call that 1. And then we're going to multiply that by 1/2 or 0.5 for wrinkled seed shape. So 1 times 0.5 is the same as 0.5. And we needed to write our answer as just a number. So since we knew that it was 50%, we just write it as 50. Okay? So that's how we do that one. Now going on to the next one. Next one's talking about cattle. This is kind of a different sort of a problem. So it says, code color and pattern is controlled by an autosomal gene locus to be contained in 1 of 4 alleles. The dominance hierarchy. Okay, when you talk about a hierarchy, you're talking about one thing is greater than another thing, which is greater than another thing. That's kind of what you're talking about with a hierarchy. And so with this hierarchy, so rather than having just a strict dominance recessive pattern where one thing is dominant to another, you have Dutch belt, which is represented by a capital S, is dominant to the Hereford genes, which is big SH, which is dominant to Sollard pattern, which is SC, which is dominant to Holstein, which is just little s. Okay, so you have more than one thing that's dominant to another thing. So the question says that we have a Dutch belt bull, so that's the dad, right? The dad's carrying the allele for Hereford, was crossed with, okay, so he's carrying the allele for Hereford, crossed with the mom is going to be a pure breeding Holstein. Okay, and then we're asked, what's the probability the offspring is going to be a Hereford? And then it says, record your answer as a value from 0 to 1. So this time we want an actual decimal as our answer. Okay, well let's look at what the parents' genes are. We see that the dad is a Dutch belt bull carrying the allele for Hereford. So Dutch belt, capital S. He's got the alleles for Hereford, so he's also got SH. So the dad is capital S, SH. The mom is a pure breeding Holstein. Okay, so she's going to have 2 little s's. Okay, so that's how the Punnett square is set up. And so we put the genes like this, and we do the cross, but we're looking for the probability that the offspring is going to be a Hereford. So again, a Hereford is going to be SH, and so the only way we're going to get a Hereford is if we get SH and then some other genes that are recessive to SH, so maybe SH and SC or SH and S. So here we see 2 situations where we have SH and S, which is recessive to it. Okay, so we've got 2 out of 4 are going to be Herefords. So what you can do at this point then, we found the probability of getting a Hereford, and so we know it's 2 out of 4, and so you would write your answer then as a decimal. You would find out what 2 out of 4 is as a decimal, and so that's going to be decimal because it goes from 0 to 1, and you round it to 2 decimal places. Okay? So that's how we do that one. Okay, next question. A recessive sex-linked condition causes hemophilia, bleeder's disease. If a normal female carries a normal male, marries a normal male, and produces a hemophilic male child, what % of all males born from this cross could be hemophilic? Okay, so that's what we're looking for, the percentage of all males born that are hemophilic. And we'll see later why that wording of the question is important. Okay, so whenever we look at hemophilia, we're looking at an X-linked trait, right? So here's how we set up the genes. This is very important. How you set up the genes for something that's an X-linked trait, such as hemophilia, which is one of the more common ones. Well, hemophilia, fortunately, is a recessive trait. It's a good thing it wasn't a dominant trait. So we call the normal trait X with a little hat and then a capital H, and the recessive trait X with a small h. Okay, and the question says we've got a normal female. So the mom is going to have X with a capital H and another X with a small h. Because it says that she produces a hemophilic male child. So if she produces a hemophilic male child, the mom must have the recessive gene, the hemophilic gene, right? If her child is going to be having the hemophilia. Okay, so that's something we can get from the question. The question says the dad is normal as well. So dad is always XY instead of XX. So he's always just got plain Y, but as far as his X, it's got to be X capital H because he's normal, right? If it was X small h, he would have hemophilia. Okay, so we know what the parents' genes are. Now that we know what the parents' genes are, we can do the cross and then find out the answer to this question here. Okay, so we do the solution here, and here's what we get. So let's go on the next page and see what the analysis of that is. Okay, so I want you to notice what the wording is, guys. The wording of this question says, what percentage of all males born from this cross would be hemophilic? So it's asking you for the percentage out of all the males. Now, if the question would have said, what percentage of all kids would be hemophilic, let's see what the answer would be. Okay, well, is this kid hemophilic? No. This is a female child, and it's got both dominant genes, can't be hemophilic. For this girl, it would also be normal because it's got the dominant normal gene. For this male, he's got the normal gene. All right, so he's not going to have hemophilia, but this one would. This male would because he's carrying the recessive gene. So 1 out of 4 kids, if the question was asking if the percentage of all kids that would have hemophilia, we would say 1 out of 4. But that's not what the question is asking. The question says, what percentage of all males? Okay, so we just look at males. We're just looking at XY, these guys here. Okay, this guy is normal because he's got the normal, capital H. This guy has hemophilia. So 1/2 or 50% of males are going to have hemophilia. So can you guys see how it's really important to focus on what the question is asking specifically? Is it saying what percentage of all kids or what percentage of all males? Okay, so you would write something different in these blanks depending on what it was. All right. So let's look at the next one. The next question kind of ties in, kind of relates back to this question. It says in question 5, what percentage of female offspring would be expected to be hemophilic? Okay, well if a female is going to be hemophilic, she's going to have X small h, X small h. And this one doesn't. She has 2 capital H's, and neither does this one. We say that this gene covers this gene, right? It protects you. Even though you've got the recessive gene, the normal gene is protecting this female here from having hemophilia. So we would say that 0% of females would have hemophilia. Okay, the next question says, what percentage of female offspring would be expected to be carriers? Well, if you're a carrier for a disease, that means you have at least one gene that is the disease trait. And that would be this one here, X capital H, X small h. So what percentage of females? Well, 1/2, right? And we would just convert that to a percent. Okay. So on to the next one. Next question says, when conducting his research into the genetics of pea plants, Gregor Mendel cross true bleeding, sorry, ever had one of those days, it's hard to talk? True breeding tall pea plants with true breeding, I've got it written twice, it should just say true breeding short pea plants, okay? We've got true breeding tall and true breeding short. Okay, and the question says the offspring of this P1 generation, in other words the parent generation, the F1 generation, that's the kids we're talking about, were interbred and produced the F2 generation. The height of each plant in the F1 and F2 generations was recorded. Okay, so we've got quite a bit of info here. The question says, in pea plants, if tall is dominant over short, which graph best represents the expected distribution of offspring in the F2 generation? Okay, so we're looking at the F2 generation. So let's figure that out. So, go on to the next page. Okay, we said that one of the parents is going to be tall and true breeding. In other words, purebred, right? So this is one of the parents, and the other is short and purebred. So it's going to have two small t's. So we know what genes the parents have, and we would cross those genes to get the F1 generation, right? So we do that. We cross the genes here, and we get the F1 generation. Every one of the kids in the F1 generation have big T, little t gene. And so that's what the F1 generation is going to look like. But the question says, if we look at the question says, what's the expected distribution of offspring in the F2 generation? So in order to get the F2 generation, we take any one of these big T, little t, and cross them with another big T, little t. Since everybody's the same, we just cross two of them. And then we'll get the F2 generation. So if we do that, if we cross big T, little t times big T, little t, we get this. Well, we can see these results are showing us that 3/4 are going to be tall and 1/4 is going to be short. Okay, so that's what things are going to look like in the F2 generation. Some kids are tall, some kids are short. So you can look at these results here, and of course this here represents a tall plant, this represents a short plant. So you can look at which answer represents an F2 generation where there's both tall kids and small kids, and you would choose that as your answer. So can you see how you get that as the answer? First of all, how you do the genetics part with the setting up the Punnett squares to get the F1 generation and then the F2 generation. Look at the results for the F2 generation and then choose which graph matches with those results. Good stuff. Okay, so on to the next question. The next question says, in Drosophila, which are fruit flies, straight wings, capital S, is dominant to curved wings, small s. A curved wing female lays 200 eggs. The eggs hatch into larvae and they mature into adults, all with straight wings. Okay, so first of all, we have a curved wing female. Just underline that. All the kids are going to have straight wings. And then it says the information indicates the genotype for the unknown male is likely. Okay, so that's what we have to find out. What do the dad's genes look like? So we need to look at what the mom's genes look like, curved wing, and we know the fact that all the kids have straight wings. Let's use that information to find out what the dad's genes are. Okay, so first of all, we know that straight is dominant. So we've got capital S for straight, small s for curved. We know that all the kids have straight wings. So what can we say at this point about the kids' genes? Well, we can say they're either two capital S's or big S, little s. We don't know yet, but you can just write that down because that might help you later on if you write it out as two big S's or big S, little s. Now, the mom is curved wing, so two small s's, and we need to know what are the dad's genes. Well, we can decide that the dad is likely big S, Because if you combine big S with two small s's, you're going to get big S, little s. In other words, that's going to give you straight wings because the capital S is dominant. Straight is dominant over curved. Okay? So we're guessing that the dad, we're making a very bold prediction, well actually we're making a reasonable prediction I should say, that the dad is big S, because that would give us kids that are going to be all straight winged. If the dad was big S, little s, then his little s, some of his little s's would combine with the mom's little s's to produce kids that have two small s's, which would then be curved. But we found out in the results, the question said the results were that all the kids had straight wings, none had curved wings. And I think we had quite a few results, right? We had 200 eggs. That's a lot of results. So we have really good reason to say that the dad is going to be two big S's, because if he had a small s, you would get some kids with the recessive traits, and you don't. So now the question says, so now that we know the dad has two capital S's, the question says, what is he likely? Is he homozygous, heterozygous? You know, is he either this or that? Well, remember what homozygous and heterozygous mean? Homozygous means that you have two traits the same, or two genes the same, I should say. So for instance, big S would be homozygous. Two small s's would be homozygous. But if you're looking at big S, little s, that's heterozygous. Okay, so you can look at the results we get for what the dad's genes are, and you can decide whether he's homozygous or heterozygous, or which one of those answers is most reasonable. Okay, so I hope you're understanding how you can look at the information in this question and kind of boil it down to figure out what the answer is. But please practice these questions over and over again so you can do it on your own. Okay, so again we're dealing with fruit flies again, and this time we've got recessive allele EB and gray body color by the dominant allele. Okay, so ebony color is EB, and gray body is going to be EB with a plus. Okay, that's sort of a different way of doing genetics than we've been used to, but we'll see how it works out. Okay, so we've got one of the things we're looking at is color, and that's with the EBs. With vestigial wings, that's a recessive allele, and it's VG. Long wings are going to be dominant, and that's VG with a plus. Okay, so the dominant trait is just shown with a plus. So, we're told that the female with ebony body color and vestigial wings is going to be mated and produce the following offspring. And the question is saying, what's the genotype of the male in this cross? Okay, well, let's first of all just kind of define what's dominant and recessive. That's always important in any genetics question to get that right off the bat. And then, you know, look at the parents that you're given and figure out what their genes are. So we're told that the female is ebony and vestigial, both recessive traits, so that's what the female's genes look like. The question says, what's the genotype of the male? Well, in order to figure out what the genotype of the male is, you always need to look at what the kids are. What are your results in the question? You know, and we're given all these results, and so we need to somehow use those results to help us figure out what's going on. Well, a lot of times you can draw conclusions based on results. Like, you look at the fact that we have 41 flies with ebony wings. We've got here another 46 flies with ebony wings. So we've got 41 and 46. You add those together, you've got 87. Then you look at the ones that are gray, so the other color alternative. What do we say, 87 for ebony? Here, we add these two together, and we're going to get 83. Now, 83 and 87 are pretty close, right? So we're getting pretty close results for color for ebony and gray, right? Then if you look at long, we've got 41 and 43, which is going to be 85. Okay, for long, we look at vestigial, and you add together 39 and 46, which is also going to give us 85. So what we can conclude is we're getting similar numbers of gray and ebony and similar numbers of long and vestigial. So in other words, the results are similar for whether we're getting the recessive trait or the dominant trait for both the color and the type of wings that we have. So that's really important. So, excuse me. So what we need to figure out, since we know this is the female, we can figure out which one of these males, these are all the genes for the males in our answers, which one of these males is going to give us a cross that's going to give us roughly equal numbers as far as the color and as far as the wings. Let's just focus on the color for the moment. So we've got EB, EB. If we want to get roughly equal numbers, the male would have to have EB plus EB. Okay, so let's see why that's going to be. So I'll just write that out, EB plus EB. Okay, so there's the male's genes. Now you can see if we're just focusing on the EBs right now, if you cross this EB plus with EB, you're going to get EB plus EB, EB plus EB. Both those two kids are going to have this dominant trait, the trait with a plus. Okay, it's going to be dominant over the genes that don't have a plus. Okay, if you cross these genes that have no plus, you're going to get EB cross with EB, EB cross with EB. So you can see that out of 4 crosses, or 4 kids produced by crosses, 2 are going to have the dominant traits, 2 are going to have the recessive. So the male is going to have EB plus EB genes. And so that's what it's going to be as far as color, and then you can figure it's going to be the same situation as far as the vestigial wings versus long wings, right? So it's going to be the same thing as far as the VG. So look for an answer where you have a male that has this as far as color and the similar sort of situation going on as far as the wings. So I hope that kind of makes sense as far as why those would be the male's genes. Based on the answers that we got the same number of ebony and gray, the same number of long and vestigial, so this kind of a cross would produce that as results. Okay, on to the next question. So this next one here is talking about one of these pedigree charts, and it's important that we first of all just review how these pedigree charts work. So first of all, remember that square stands for male and circle stands for female. That's any pedigree chart. You can always remember that's always going to be the case. The other difference, the other thing to note is that dark always stands for having the trait. So whatever the trait is that is being discussed in the question is going to be dark. So dark means you're affected by the trait. Okay, so here it's talking about opalescent dentin. So anyone who's dark means they have opalescent dentin. Anyone who's white is not going to have that. Okay, so the question says, this pedigree shows the incidence of opalescent dentin in the family. People with this trait have insufficient enamel on their teeth. The dentin inside the teeth shows through, giving the teeth a mottled pearl-like appearance. Okay, so what is the question asking us for, now that we know how pedigree charts work? It says the pedigree indicates the trait for opalescent dentin is controlled by a gene that is, okay, so we want to know what the pattern is that's going on. Is it an X-linked pattern? Is it a dominant pattern? Is it a recessive or a codominant pattern? Well, let's see how we can figure that out. So we'll go on to the next page. What I would do, if I was trying to figure this out, I would come up with a theory. I would test all the different theories. So test, first of all, the theory that opalescence is a dominant trait. Then test the theory that it's a recessive trait, and see what kind of answers you get. See if they match up with what you're seeing going on in the chart. Okay, so how are we going to set up the genes if opalescent was the dominant trait? Well, if it was dominant, we would say O equals opalescence, right? Because we usually take the first letter of the trait that's going to be the dominant trait. And we would say small o is non-opalescent. Okay, so let's match that up against the results that we get. So you can choose any area of the chart and kind of examine the results. I happen to think that this was kind of interesting, this area right here, looking at these parents and these results, because we can see we have two parents that are affected, and we get kids, some are affected, some are not. Okay, so we need to ask ourselves, is that a dominance pattern going on or recessive? Well, sometimes you might recognize that right off the bat. But if you don't, then set up a Punnett square and test it out and see what you get for results. Okay, so we know that we're going to have some kids having recessive genes if the parents are going to produce kind of a mix, right? So if we're assuming that opalescence is a dominant trait, if it's a dominant trait and then these parents have the genes, then these parents would either be two big O's or big O little o. But we're thinking that they would be big O little o rather than two big O's, because some of their kids would have the recessive trait, some would have the dominant trait, right? We've got a mix of kids. So those would be the parents, and then we see that we would get kids, some of who would have the recessive trait, two little O's, and some would have the dominant trait. So our results here are matching up with the dominance pattern. We're seeing some of the kids have the dominant trait and some have the recessive trait. So far our theory that opalescence is a dominant trait is holding up. Let's test out some other different kind of patterns and see why they would work or not work. So here's the theory that opalescence is a recessive trait. Now if opalescence was recessive, then we would say N equals normal, capital N equals normal, okay, because we always take dominance with the first letter of the dominant trait. Okay, so small n would be opalescent. So we want to see if this is holding up. Okay, so if opalescence is a recessive trait, well, again, examining the same area of the chart that we did in the last time, these parents would have the recessive trait, right? If opalescence is recessive, they're affected. So they would have two small n genes, each of them, right? Now if the parents had two small n genes, then the question I have for you guys is, how would you get kids having both dominant and recessive traits if you have parents that only have recessive traits? You should have kids that only have recessive traits, but here we see kids having both dominant and recessive traits. So in other words, the chart is not matching up with the theory that opalescence is a recessive trait. Okay, so let's try out one more theory. One more theory is that opalescence is an X-linked trait. And so how does X-linked work again as far as the genes? Well, let's look again at hemophilia, which we already looked at a little bit earlier in another question. And we said that X capital H was normal, X small, I should say, H, is hemophilia. Okay. Now what happens when you do that kind of a cross? Females, as I mentioned earlier, the recessive gene is covered in a female. As long as you get one dominant gene, the female is going to be normal. Whereas any male that gets the recessive gene is going to be hemophilic. Now, in other words, with males, the recessive gene is not covered by the normal gene. So what that tells you also is that with an X-linked gene like hemophilia, you're going to get more males being affected than females, aren't you? Because females getting the recessive gene, as long as it's covered by the dominant gene, they're going to be normal female. So you should be able to look at the chart and see more males having the affected trait if this is an X-linked trait. So if we look at the chart here, we should see more squares being colored in if we've got an X-linked pattern going on. So do you guys see more males with the square being colored in than circles colored in? I don't think so. Okay, so we would say this is not an X-linked pattern. So we would conclude that the first theory we tested out, that was a dominant pattern, because when we did the genetics in it, we tested it out and it worked, we would conclude that it's a dominance pattern going on. Okay, so can you see how you can check out these pedigree charts and see what type of pattern is going on? Now the next question here is relating to that, and it's saying if you have two individuals from this chart having another child, what's the probability this child would have opalescent dentin? Now this question is assuming that you know what type of pattern is going on. So since we decided that it's a dominant pattern going on, we are going to use the genes that we came up with when we talked about the dominant pattern in question 11. And what we're going to do, this question says individual 3-1 and 3-2 are having a child. Okay, here we see row 3, and here's individual 1, here's individual 2. Those are the two kids we're talking about having another child. So we need to figure out what their genes are based on what genes are for the dominance pattern when we looked at it in question 11. So find out what those genes are for these two parents, do the cross, and then figure out what the probability will be that the child will be a female with opalescent dentin. So again, if we're going to figure out that probability, we just look at the we're just looking at the females, right? Okay. So we want to record our answer then as a value between 0 and 1, some sort of a decimal. All right. So we're looking for the chance of being female with opalescent dentin. So on to the next question. Okay, the next question says, a sex-linked disease that can cause fetal bleeding is, so which one is it? Hemorrhage, Tay-Sachs disease, sickle cell anemia, or hemophilia? Well, I'm actually not going to answer this question for you guys, but I want to review these different diseases, and I'll let you guys decide which disease is the disease that causes fetal bleeding. Okay, so I'm just going to review these here, and I want you guys to know them well enough so that you can match each one of them. Okay, so first of all, a disease caused by an abnormal form of hemoglobin, that one is going to be sickle cell anemia. Okay? And the next definition we have, a sex-linked disease that causes fetal bleeding and follows an X-linked inheritance pattern, homozygous individuals are dominant, that's hemophilia. Next definition, this disease involves a gene mutation and does not follow an autosomal recessive inheritance pattern. Okay, so that's Huntington's disease. And I said, remember, this is a degenerative disease of the nervous system that affects muscle coordination and some cognitive functions, in other words, brain functions. Okay, next one says a disease that involves a gene mutation and does not follow an autosomal dominance inheritance pattern. That one is going to be Tay-Sachs disease. Okay, so again, you've got a similar definition going on here for the last two. So this Tay-Sachs disease involves young children and results in both mental and physical deterioration. Most children die by 5 years old. Okay, so know each one of those diseases, definition. Okay. I think we're getting close to the end. We're in question 14, and we've just got a little more to go. Okay. So, Thomas Hunt Morgan bred a normal red-eyed female fruit fly with a white-eyed male fruit fly. The offspring in the F1 generation were all red-eyed. All the females in the F2 generation had red eyes. 1/2 of the F2 males had white eyes. This shows that in fruit flies, okay, so we've got different possibilities going on. It's asking us whether the gene for white eyes is recessive or whether it's dominant. Is it on the X chromosome? Is it on the Y chromosome? So let's first of all look at an overall description of the problem. I find sometimes just kind of setting up a diagram for certain questions like this one help us to keep track of what's going on. So the parents, we have a red-eyed normal female crossed by white-eyed male. In the F1 generation, we got all red eyes. In the F2 generation, all the females had red eyes. 1/2 of the males had white eyes. So we need to figure out whether it is on the X chromosome or Y chromosome, whether white eyes are dominant or recessive. Well, what I would do first of all is assume that it's on the X chromosome rather than the Y chromosome. In this course so far, we've only looked at X-linked traits. We've never looked at Y-linked traits. We just looked at ones carried on the mother's chromosome. So let's assume that for the time being, and then look and see if the information kind of bears that out. So we're going to assume it's an X-linked trait, and we just need to find out whether white eyes is recessive or dominant. Okay, so we're going to test the theory that white eyes are dominant against the theory that white eyes are recessive. So again, just like earlier questions, set up the Punnett squares, test out these theories, see if it holds true. So our first theory is white eyes are dominant. Now if white eyes are dominant, if the mom has red eyes, she's going to have X with 2 small W's, right? Because we're saying white is capital W for white eyes, right? Small w for red eyes. Okay, so again, notice this is the way we set up the genes when we're talking about an X-linked trait, right? That's what we're assuming. Just like with hemophilia, we had X capital H, X small h. Here we have X with a small w. Okay, so that's the mom's red eyes. The dad has white eyes, so he's going to be X capital W if white is dominant, and he's going to have a Y. So we're going to do the cross here, and we're going to find out what we have. So let's look on the next page. So the question is, in the problem we were told that the F1 generation was all red eyes. Is that what we're getting with our results? Remember, red eyes, like the mom, was when we have 2 small W's? No. We have some who have the capital W, the dominant trait. These are going to be white eyes. So we don't have the F1 generation showing all red eyes. So because of that, our theory that white eyes is dominant goes out the window. So we have to test the other theory, that white eyes is recessive. Okay, so if white eyes is recessive, then we define, instead, see, we're setting things up differently here, right? Instead of having W's, we're having R's, because red is dominant, so we're going to have X capital R for red eyes. That's for the mom, and we're going to say it's X little r for the dad, because he has white eyes. All right? So we have these genes, and so we're going to do the cross and see what the results are. Well, when we look at the results, remember red eyes are dominant, that's a capital R, we see that every one of the kids are going to have red eyes. So this is showing us that the statement that the F1 generation all have red eyes is holding true. So we say the theory that white eyes is recessive is a correct theory. So we're saying we've got X-linked genes. We made that assumption. And white eyes is recessive. That matches up as well. And so we're saying that it's going to be white eyes being recessive. So we'll look back at the question and see which answer we would choose then. Another thing I want you guys to notice, we said that in the F2 generation, we've only done the Punnett square for the F1 generation, we said that in the F2 generation, all the females are going to have red eyes, so they're all going to have the dominant trait, but the males, half of them are going to have the recessive trait. So does that make sense with an X-linked genes that more males than females are going to have the recessive trait? Well, it makes sense when you think back to hemophilia. We saw that more males than females had the recessive trait, hemophilia. Because with the females' genes, again, it was covered by the other trait. Remember, we had X with a big H and X with a little h. So the big H was covering the little h, and so the female didn't get hemophilia. Okay? So this again makes sense. This statement in the question is matching up with everything as well, because it's saying that half of the males are getting the recessive trait, whereas the females are not. So again, it's making sense that it's an X-linked trait, based on what we know about X-linked traits. So when we would go back to this question, what it was asking in the beginning, it says, is it going to be on an X chromosome or a Y chromosome? We assumed it was an X chromosome, and now we can answer whether the white eyes is recessive or dominant. Okay? So, I believe that might be our last question. And it is. Good stuff. All right, so guys, again, I can't urge how important it is to watch these recordings, to practice these questions over and over again. I would practice them enough that you know how to do all of them really well without looking at the answers. If you do need help with any of them, give me a shout. But I believe that going back over the recording, watching it again, stopping it at certain points if you need to, sort of things, so you can practice things, then playing it again. If you do that, you guys can get these answers. And so again, I want to wish you guys the best on our midterm on Wednesday. And again, we've kind of divided up the genetics. So we've got half of it on Wednesday. We're going to go into more genetics later on. But let's focus on this for now and doing really well on this. And then we'll focus on the second part of the midterm, which is going to come in about somewhere in the next 2 weeks or so. So guys, God bless. Keep working really hard. And I hope you have a great day. Take care.